Hello, hello các bạn.
Hello. Thì có số điện thoại Mỹ không thì cho limit. Sáu tám linh trích số đây tiền
Sốc tiền quá rồi mà. Mua cái nữa. 60 ngày chắc đủ.
lần cả năm luôn.
Muốn đặt thì đặt thêm 2 lần, mỗi lần đặt được 5. Kéo ra làm 1 ca mua được 4 tháng. Cứ để đấy, 2 thằng nữa 4 năm tiếp rồi.
Okay, so good morning everyone and welcome back to my course, Bioinformatics. So for the students have already involved in the bioinformatics practical assignment, please remember that when you submit your assignment, it has to show up the text submitted for grading in green. Otherwise, it will stay at the draft status, and if you stay at the draft status, it will be not count that you have already submitted your assignment. Okay, so remember. So with this 23 students who still in the draft submit, I still open the deadline, so you can submit it later. But remember to submit it, not put it in the draft form. Okay? Okay, so today we will get into the chapter 2 related to, we will discuss with each other on the mechanism how DNA was replicated during mitosis and meiosis. So, first talking about cell division. So the development is a miracle of life when an embryo embryonic cell, the first stem cell, or sometimes we call user zygote. This zygote will develop from one cell stage divide fireplace and one fireplace. The zygote, the first stem cell, has a life cycle around 24 hours. That means in each 24 hours, it will divide one time. So after one day, we will have, it divide one time, we will have 2 cell stages. After the next 24 hours, we will have the 4 cell stage. And after the next 24 hours, after 3 days, we will come to the 8 cell stage. So estimate the time of cell division is really important, especially when with the technique to develop the embryo in the birth assistant. So for example, many hospitals and many institutes have developed a service to raise embryonic development for the couple that have a difficulty in giving birth in a natural way. So they will put the zygote, is a combination of a fertilizing egg into a bed cell to raise it, and after they will calculate to estimate the cell cycle after 3 days, that means in the 8 cell stage, or in 5 days, that means in 2 power with 5, that means 32 cell stage. To make a genetic test to check that if the embryo have any genetic abnormality. So as you can see here, it's a time lapse of zygote undergo several cell stages. There is also a technique to estimate that if the embryo development normally based on the estimate time. For example, if the cell, the zygote, develop in 24 hours and divide one time, that's normal. But if divide in 16 hours or divide in 30 hours, that means too far from the normal time lapse 24 hours, that so abnormal. And also, if all the cells are not divided equally, some cell is smaller, some cell is larger, or some, for example, some cell is divided later than others. So we have observed sometimes not 2, not 4, not 8, but like 5 cells or 7 cells. That means something abnormal will happen. And when they make a genetic test with the embryo, say that they see that there is a significant increasing of genetic abnormal on those cells. In later of this chapter, we will discuss about the cell cycle and about the genes that control the cell cycle. A small demonstration how really.
kêu mà có ngủ hay là mấy thôi.
thầy cho kiểu nghe YouTube ấy, tại vì một tiếng thầy nghe
Mua cho các bạn. Vì vậy thành ban đầu đúng nhiều quá. Người ta lên có sự đấy mới làm.
bạn đau lên thì là phải có sợ gì nữa.
Alright, so during development, at early cell stage, we will have two layers of cell. The outer layer cell and the inner layer cell. The outer layer cell later will develop into the placenta, and the inner cell will develop to an individual. So that's why the genetic background, because all of this, both the outer cell and the inner cell was developed from one cell, the first stem cell, the zygote. So let's say that all the genetic background, all of these cells, all these cell clusters are the same. That's why during around 6 weeks to 8 weeks of the embryo development, the medical doctor can have a relance of several techniques to run a genetic test using only the outer cell. Because in general, all the mutation show up in the placenta will show up also in the inner cell, in the individual later. Okay? Now, fundamental question. Do you know how embryonic age was calculated? Sim 1, the embryonic age was calculated. Let's calculate us when the sperm mediate and fusion afterward the fertilization. It's calculated since the zygote starts to divide. So when your mom or your sister who are pregnant and went to the medical doctor to regular visits, regular check-in, they will provide result that he or she will have a baby in the age of the embryo is like 7 weeks or 8 weeks. So 7 weeks or 8 weeks, see what? See what event? Ngoài biết
luôn là biết là được.
Không ai biết tuổi thai tính như nào.
Phổ cập thế thầy ơi, em chưa đẻ bao giờ.
Chết. Kiến thức cơ bản này chết.
Đã đẻ bao giờ đâu ạ?
Mấy điện rồi.
Thế tuy nhiên là chưa đẻ bao giờ, nhưng mà khi có người thân đi khám đúng không? Ví dụ là chị Quỳnh hoặc là cô hoặc là ai đấy đi khám thì bác sĩ cho mấy kết quả là tuổi thai được 6 tuần hay 8 tuần. Thì 6 tuần hay 8 tuần đấy tính từ thời điểm nào? Okay, so that's the first answer is calculate from the sperm start to meet the egg. Anyone have other answer? Yeah.
Count from the last day
It calculates from the last day of the period. So, what event happened? What kind of event happened when the last day of the period? No, not exactly. So, actually, the right answer is it counts from the last day of the period. But the last day of the period is when the egg cell will mature and have enough ability to develop into the embryo. But it has not met the sperm yet. Okay? Cho nên là tuổi thai được tính là từ ngày trứng dụng lần đầu tiên, chứ không phải là tuổi thai được tính từ ngày trứng được tinh trùng nhé. Nên là sau này ai mà đưa lập gia đình xong đưa vợ đi khám tính tuổi thai 6 tuần, đấy đang đi công tác hay là đang đi đâu đấy. Đấy thì đừng có nghi ngờ gì nhé, vì là tuổi thai là được tính từ khi trứng bắt đầu dụng. Trứng khi bắt đầu dụng có thể là sẽ gặp tinh trùng sau khoảng độ 5 đến 7 ngày nữa, không sao cả. Nhưng cái tuổi thai là được tính từ khi trứng bắt đầu dụng. Cái quan niệm phổ biến sai lầm phổ biến đó là từ tuổi thai tính từ khi trứng bắt đầu gặp tinh trùng, đấy không chính xác. Cho nên cái ý nghĩa của cái kết quả khi mà các em sau này nếu các em có đọc được thì là ý nghĩa là như thế. Tuổi thai là 6 đến 8 tuần là tính từ khi trứng bắt đầu dụng. Ok chưa? Okay, so today we will discuss on cell divisions into fundamental process in the cell. The mitosis will happen in the somatic cell and in the gamete producer cell in the early stage only, and the meiosis in the mature gamete producing cell. So we will have in the cell cycle or in the when the cells divide, of course, it has to develop all the organelles, it has to duplicate everything, and among those have to duplicate there's DNA. So DNA have to be replicated as all the other organelles like the microtubules, the mitochondria, the nucleus regions have to be copied before the cell divides, or the cell divide is also used as a term cytokinesis. So, in the mitosis, we will have four main phases: the prophase, the metaphase, the anaphase, and the telophase. And the interphase, the most longest phase is the interphase, was not counted as mitosis, although some books refer that the interphase are the part of the mitosis, but this information is not really accurate. So officially, we have only four phases in mitosis. So behind the mitosis, we will have three other phases: the G1 phase, the S phase, and the G2 phase. So DNA was replicated during the S phase. So back at the time, let's go back a little bit in history when people find out about the DNA structure in 1953 by the X-ray experiment by Franklin. So they wonder, with this helix structure, how a DNA molecule can be replicated. It can be replicated. There's three possibilities that this one can be replicated from the scientists at the time. First is a semi-conservator, which is followed by Watson and Crick, that from original DNA strand, double strand, we will have each strand will separate, and the DNA replication complex will use each strand as a template to synthesize a new DNA strand following the complementary rule, Chargaff rule. A is linked with T and G is linked with C. And so on to the next replication. The other one, the octary, is calculated as a conservator. That means that one DNA strand could be using entirely the two strands as a template to synthesize the new DNA strand. So as a result, we will have this DNA strand the whole two strands as template to synthesize a new one. So after the first replication, we will have two double strand DNA, which one is using the whole two strands from the original one, and the other one is brand new. And so on to the next replication. The third theory, the third hypothesis, is that this process will depend on the regions that the DNA replication will use. For example, with this one is when you these regions as a template to synthesize the new one, and with this one it will use this one. So after first replication, we will have like what we use the word chimera, a chimera state of DNA, when part of the DNA fragment was from the original one, and part of the DNA was brand new. So, with these three hypotheses, scientists have been designed an experiment to prove that which theory was accurate at the time. So, in 1957, Mansfield and Stark have designed an experiment with radioactive nitrogen. So we have two nitrogen, the nitrogen 14 and the nitrogen 15. So because nitrogen is important, is one of the part that located in the base matrix, one of the important part of the nucleotide, which is the monomer of DNA. So the experiment is designed like this. First of all, we grow E. coli bacteria, a prokaryotic cell that is very popular in biological experiment back then and even now. So we grow it in the regular nitrogen medium. So this is not radioactive. We extract the DNA from the cell and centrifuge, and we obtain the normal and regular DNA. The DNA was commanded by the N14. Okay, so this one is regular, not radioactive. So we pick another E. coli. We will grow it in the medium contain only the nitrogen 15, which is radioactive. Extract DNA and obtain the DNA strand with nitrogen 15. Okay. So now we will pick one of the cell in nitrogen 15 and transfer it into the medium contain only the normal nitrogen, the 14 nitrogen. Let it replicate and divide for a couple of times. As I remember, the cell cycle of E. coli are around 40 minutes to 1 hour. So in each 40 minutes or 1 hour each will divide once. That is very quickly accurate rate of the cell division. Just compare that in human stem cell, which is the most fast device cell in human, and it will take 24 hours for a cell cycle. We will talk about it later. So when we pick up the cell and put it to the nitrogen 14, and we extract the DNA, centrifuge it. So at the time when we centrifuge it, the centrifuge, we put a centrifuge force on the medium. So the DNA, the molecule that heavier will be located in the bottom, and which molecule was lighter will show up in the top regions. Okay. So in here, after the first subdivision, because we estimate that the E. coli will divide one in 40 or 45 minutes to 1 hour. So after 1 hour, we centrifuge it and we see that the DNA was located in the middle. Okay, because not any double strand was really heavier than anything, because in both strands, double strand, we want one strand with nitrogen 15 and one strand with the nitrogen 14. So we let it divide another time. So we wait another 40 minutes to 1 hour. So the E. coli divide a second time, and then we centrifuge it again. So this time we will have two strands. One with the 15, 14, the same with the one in here, and another one with the 14, 14. Okay? So, you listening to the three hypotheses, you observe the result, the experiment, what kind of conclusion we can have in here? That's critical thinking. That's the ability that I hope you will gain in 3 years of university. You observe a phenomenon, you observe a data, you provide a hypothesis. After you have a hypothesis, you design the experiment to prove your hypothesis, to know that if your hypothesis is true or wrong. And then based on the result of the experiment, you draw out the conclusion. You have 3 hypotheses. So you have the 3 hypotheses in here, the semi-conservative, the conservative, and the dispersive. You have known the experiment which is designed already for you. You observe the result. What kind of conclusions that you can have in here? Anyone? So the conclusion in here is when we observe after the second division, we have a band of 14 and 15 degrees corresponding to the band that show up in the first subdivision. And we have another strand of band is 14 and 14, which show up in here. So which theory is right? Among the 3 hypotheses.
Nhìn thấy Nguyên nó phải chào 7 kiểu đấy.
Semi-conservative. Conservative. And dispersive. Okay, so after the first subdivisions, we have 2 bands.
Chạy đấy.
Alright, so after the first subdivisions, we observe only one band is 15 and 14. And after the second subdivisions, we observe 2 bands, 14, 14, and 1 band, 14, 15. Okay? So that means the semi-conservative theory was right. But let me ask you some questions. What result from this experiment? What result you will observe if the conservative theory was right and if the dispersive theory was right? You understand my question, or you don't understand it at all? Ok. Bây giờ có 3 giả thuyết cho cái việc là DNA được nhân đôi như thế nào. Giả thuyết đầu tiên là bán bảo toàn, đúng không? Có nghĩa là DNA sẽ sử dụng một mạch. Đây, sẽ sử dụng một mạch gốc từ DNA của bố mẹ để tập hợp nên cái mạch DNA mới. Cái thuyết này thì ngày nay đã được chứng minh là đúng, nhưng giả sử bây giờ hãy giả sử mình là những nhà khoa học thời đó, mình chưa biết thuyết nào là đúng cả, đúng không? Thuyết thứ hai là thuyết bảo toàn, có nghĩa là toàn bộ cái mạch, cả hai mạch DNA này sẽ được sử dụng để tập hợp nên mạch DNA mới. Cho nên là ở cái mạch mới thì chúng ta sẽ có cái mạch cả cái mạch đôi này là từ cái mạch cũ, và cả cái mạch đôi này là mạch mới tập hợp. Và thuyết thứ ba là tùy vào từng đoạn, có đoạn thì sử dụng mạch này là mạch mới, có đoạn thì nó sử dụng cái mạch kia là mới, đúng không? Được chưa? Có ba giả thuyết như thế. Giả sử mình là nhà khoa học thời đó thì cũng chưa biết gì về công nghệ giải trình tự, chưa biết gì về nguyên tắc đoán bảo toàn, chưa biết gì. Thế bây giờ mình phải chứng minh như thế nào để biết được trong ba cái thuyết này thuyết nào là thuyết đúng. Thế thì ở đây thí nghiệm đã được thiết kế sẵn, giờ mình không cần phải thiết kế thí nghiệm nữa, đúng không? Thiết kế thí nghiệm là cái không quan trọng nhất, để đã thiết kế sẵn cho mình rồi. Bây giờ có hai loại nitơ đồng vị, hai loại đồng vị nitơ, một loại đồng vị nitơ 14 là cái nitơ bình thường, 70% trong không khí, đúng không? Nitơ thứ hai là đồng vị phóng xạ là nitơ 15, cái này khác là phóng xạ, cái này có chu kỳ bán dạng. Bây giờ thí nghiệm đầu tiên là người ta cho các em E. coli nó sống trong cái môi trường có chứa nitơ bình thường, đây là control, thì nó sẽ sinh ra cái mạch DNA chứa nitơ 14 bình thường, mạch đôi là chứa 2 phân tử nitơ, ok chưa? Một thí nghiệm khác người ta lại cho con E. coli đấy sống trong môi trường mà chỉ chứa nitơ đồng vị phóng xạ 15 thôi, nó sinh ra cái nitơ đồng vị 15 đây, và sau đó người ta ly tâm thì các em biết rồi, nó sẽ có cái lực ly tâm, đúng không? Cái gì nặng thì nó chìm xuống dưới, cái gì nhẹ thì nó nổi lên trên, đúng không? Thế thì sau khi ly tâm thì cái vạch nitơ 14 thì nó sẽ ở đây, vạch nitơ 15 nó ở đây, chưa? Cái con này chứa toàn bộ nitơ, DNA là nitơ 14 thì nó ở đây, con này chứa toàn bộ DNA là nitơ 15, nó nặng hơn, nó chìm xuống đây, được chưa? Rồi, bây giờ cho con nitơ sống trong môi trường 15, pick một con, cho vào cái môi trường bình thường nitơ 14 để nó phân bào một lần. Tôi vừa nói rồi đúng không? Chu kỳ phân bào của E. coli thông thường rơi vào khoảng 40 phút đến 1 tiếng. Thế thì sau 40 phút đến 1 tiếng thì cho 1 tiếng đi, nó phân bào một lần rồi. Tách nó ra, lại ly tâm thì lúc này tôi thu được cái mạch ở giữa. Một mạch nitơ 15, một mạch nitơ 14 vì nó phân bào một lần mà, đúng không? Và nó sẽ tạo thành hai cái mạch ở đây. Ở giữa nó không nổi hẳn như 14 mà 14 nó không chỉ hẳn như 15, nó ở giữa. Lại tiếp tục cho một chu kỳ phân bào nữa, lại đợi tiếp 1 tiếng nữa thôi. Lại ly tâm thì lúc này thu được hai vạch, một vạch là 14, một vạch là 14, 15. Như vậy là thuyết là bán bảo toàn semi-conservative là đúng. Tuy nhiên bây giờ câu hỏi này, nếu giả sử thuyết này không đúng mà thuyết này đúng thì kết quả toàn thời lượng sẽ như thế nào? Hiểu câu hỏi không? Tôi lặp lại đúng thí nghiệm này, nhưng nếu cái giả thuyết này nó mới là đúng này thì kết quả tôi nhìn được sẽ như thế nào?
Toàn đỏ toàn xanh rồi.
Hả?
Có hai vạch nó toàn đỏ hoặc toàn xanh rồi.
Hai vạch nó sẽ sao?
Toàn đỏ.
Bây giờ nếu mà chúng ta đúng là bán bảo toàn thì
cái trên cùng, cái dưới cùng.
nếu cái này là đúng thì ngay chu kỳ đầu tiên tôi đã thu được hai vạch rồi, một vạch 15 và một vạch 14. Đúng không? Thì chu kỳ tiếp theo thì chỉ có cái vạch 15 này nó càng 15 nó ở dưới, 15 nó nặng hơn mà, đúng không? Những chu kỳ tiếp theo tôi vẫn thu được hai vạch, chỉ có cái vạch 15 càng ngày nó nhạt đi, và vạch 14 càng ngày nó càng đậm lên thôi, đúng không? Thế còn nếu cái giả thuyết này là đúng
thì kết quả sẽ như thế nào? Kết quả sẽ như thế nào? Nếu cái mạch này mà đúng thì lượt đầu tiên tôi vẫn sẽ quan sát được là một mạch 14, 15. Thì DNA nó mix lẫn với nhau mà, đúng không? DNA mix lẫn với nhau. Nó vẫn là một mạch là cả 14 cả 15. Và những lần tiếp theo tôi vẫn chỉ thu được là 14, 15. Đúng không? Chỉ có dần dần hai cái mạch này nó sẽ tách nhau ra, thêm những cái mạch nữa ở đây, chứ không thể sau chu kỳ đầu tiên nó tách ra thành mạch 14, 14 và 15, 14, 15 như thế này được. Bởi vì cái thành phần 15 thì càng ngày nó càng ít đi, nhưng mà nó ít đi dần theo mỗi chu kỳ, đúng không? Còn cái mạch 14 thì càng ngày nó tăng lên, thì cái mạch 14, 15 này vẫn có, nhưng dần dần nó sẽ có những nhiều cái mạch nó bắt đầu chuyển, đúng không? Okay, so let's walk again how the Manson and Scott was integrated.
The Messelson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So the semi-conservative had been proved that is right compared to the two arbitrary. So right now we will have the parallel DNA strand. We use each strand as a template to synthesize the newly one. So as a result, in the two daughter DNA strands, we will have one strand belongs to the parent strand, and one strand is newly synthesized. Okay. Yeah. So, what is component, what is complex involved in this process? So, actually, involved in the DNA replications, we will have different types of complex that involved in this one. Different many kind of enzyme involved in this one. So in here we will have the enzyme participate in this process. We will have helicase, which is involved in separating the DNA and keep it untwisted under the release it from the double helix structure. It will unwind the DNA by cutting the hydrogen bond located in the cell. We have another helicase here. We will unwind the double helix structure by cutting the hydrogen bond. We have primase, which is the primer into template strand, and because thanks to the primer, the DNA strand could be synthesized. The main point, the main play is the DNA polymerase, or sometimes we call DNA polymerase 3, which is synthesized the newly DNA strand by keep adding the nucleotide into the new strand. We have another DNA polymerase, but this one is not like the polymerase, but like the eraser, which is the exonuclease activate that delete the RNA primer and then replace the RNA primer by the DNA oligos. And last one, we have the ligase, which is seal the DNA strand together. So, in here we have the synthesized was happened only on the one strand from 5' to 2'. So we have a double strand DNA here. First thing we have to know is the new DNA strand. Is always synthesized from 5' to 3'. So we have the first one is helicase with putting in here. Which is break down the hydrogen bond. Break. That link up to DNA strand. Okay. To separate it into single strand here. Secondly, we have another, which is primase. I have the color chocolate here. Only the white chocolate. Right. So we have the primase. We can ask the RNA. Remember, the primer here one is a primer, and it's made from RNA, not DNA. Okay? What is differences between RNA and DNA? One is a ribose sugar, and one is a deoxyribose sugar, right? So what is the differences between the ribose sugar and the deoxyribose sugar? In carbon number 2, deoxyribose sugar have the hydrogen atom, and in ribose sugar we have the hydrogen, the OH group, right? So the prime numbers are in here. So this one is 5 prime to 3 prime, and this one the 3 prime to 5 prime.
Có thức đấy.
So after we are primary here,
Bị yếu rồi ạ. Không ghi ra kỳ
we will have the DNA polymerase 3. We start off the DNA strand from the 5 prime to 3 prime, because new DNA strand only synthesized from 5 prime to 3 prime, not in the 3 prime, not in 3 prime, 5 prime. Remember this. Okay? So, because the top strand is from 5 prime to 3 prime, and the primates would already be here, so it will synthesize from 5 prime to 3 prime here. So we call it the leading strand. Because this process was happening continuously. Now in the other plant, we will have, this is in the other direction, 3 prime to 5 prime. So when the RNA primer here, so this will be the 5 prime, and this will be 3 prime. The DNA polymerase will start in this direction, right? So it will add another here. You have another DNA polymerase will add into synthesize. So we see that this process was happening this continuously. And that's why we call it the loving trend. Loving trend again. So, of course, DNA polymerase, like other machine, like a photocopy, sometimes it had an error. Normally, the error was happening in the high level organisms is less than the low level organism. To compare, for example, in bacteria, the rate of mutation was happening in H1 10 power by 7, that means in 10 million base pair, we will have a mismatch. We will have a fold of replications. In human, for example, we will have a rate about 10 power by 8. That means if 100 million nucleotide, we will have a mismatch. But thanks to the DNA corresponding, thanks to DNA tracking copying fold, we can decrease it 100 times. Drop down the fold rate from 100 millions to 10 billion base pair. We will have one mutation. But 10 million base pair, one mutation. Meanwhile, the human genome is 3 billion base pair. So that means in around 3 generations, 3 cell division, we will have a mismatch. Okay, we will have a mutation. And as you can calculate, the more cell was divided, the more cycle cell will divide, the more mutation we will accumulate in our genomes. But talking about mutation, we will have another chapter number 4, related to mutation. But for the moment, just remember that mutation is normal. We all have it. Okay? You all have the mutation newly compared to the genome of your parent. Okay? Because of the fold rate. Even when the body have mechanism to refix that.
Cells, like these prokaryotic E. coli cells, replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of 2 strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp motor, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase, because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
So during the process, this discontinuously fragment we call the Okazaki fragment. And lately we have again DNA polymerase, but this one is DNA polymerase I. Remember that DNA polymerase have two characteristics. First is polymerase. That means adding nucleotide. When in the environment they are full of free nucleotides. And the second one is isonuclease. That means deleting. The RNA polymerase when there is no nucleotide in the environment. So, the DNA polymerase will come and erase all the RNA primers and replace it with the DNA. This times isonuclease activity to replace RNA primer by DNA fragment. And last, we have the ligase. Join up the Okazaki fragment. Together to form a continuously thread. Okay. So let's come with the first process today is mitosis. So mitosis is happen in most of the somatic cell and also in the gamete cell, sex cell, in early stage of the sex cell when it needs to divide in the divide not to produce gamete but to produce more gamete production cell. Okay, the immature gamete production cell. So mitosis begin after the interphase. Okay, interphase not count as a part of mitosis, but during interphase, all the ingredient, all the DNA material like DNA and other organelles was duplicate, prepared for a mitotic phase. So after the interphase, mitosis involve only 4 phases, which is prophase, metaphase, anaphase, and telophase. So in prophase, we have the membrane, nucleus membrane will disappear. The chromosome was paired up, the sister chromosome will pair up and attach to each other in the centromere. And if began to twist, and so we can easily observe it under the microscope, but it will be twist maximum in the metaphase phase. So we have also the duplicate centriole, which is moving in the 2 parts of the cell, and it starts to producing the microtubule and start to line up the chromosome under the metaplate in the middle of the cell. So in the metaphase, we will have the chromosome was located in the metaphase of the cell already. And at this time, metaphase, the chromosome was twist maximum, so we can observe it under the microscope. In the end of metaphase, the microtubule of 2 centriole here start to separate the 2 sister chromosomes together and start to pull it up in the 2 directions of the cell, like in the anaphase here. Okay. So in the other phase, we will have a telophase when the 2 chromosome set was formed up and the nucleus membrane was reformed again to separate the 2 cells together, and the 2 cells start inform the cell membrane to separate the 2, which is cytokinesis.
adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or GAP1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or GAP2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the 2 identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all 3 phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
So like we have discussed, DNA will replicate, the machine replicated, sometimes it makes mistakes. And when mistake in DNA replication happen, mutation will appear. Okay? So this is a rate of mutations. So normally the higher organism, higher level organisms, the less mutation, less mistake it will make in the DNA replication. Of course, based on to the structure and based to the stability to the environment, RNA is less stable compared to DNA. So with the organism we have RNA genome, normally the mutation rate are much higher compared to DNA. For example, DNA virus will have a mutation rate from 10 power by minus 6 to 10 power minus 8. That means in each 1 million base pair to 100 million base pair, it will make a mutation. Okay. RNAs will have a much higher rate, which is around 1,000 to 100,000 mutations per base. So that explains why all the RNA virus will have many variants like the coronavirus. Okay? Coronavirus is a retrovirus, the RNA virus. And because it's composed the genome of RNA, so that we have many variants of coronavirus. We have now this type C, type Odera, right? Many types of mutation is made to the treatment. The diagnostic of retrovirus is much more complicated than to the DNA virus. So in humans, the mutation rate is from 1 per minus 8 per site per generation. But because we have a mechanism to fix this, it will drop down to around 1 million base pair, 10 billion base pair for mutation. So with this high mutation rate, what is the mechanism to control this? So we have, like in the clip we see, we have a mechanism called cell cycle checkpoint. There are many cell cycle checkpoints, but we can name three main cell cycle checkpoints. That's the checkpoint in G1 phase between the G1 controlling from G1 to the S phase. We have another checkpoint in the G2 to N phase. And the last one is the metaphase checkpoint. So, in here, for the first checkpoint from G1 to up, it will check for if the nutrient is enough, because the S phase is the phase when DNA was synthesized, okay, doing DNA replication. So it will check if the nutrient or the ingredient, the nucleotide, the energy is enough for the DNA replication process. All the growth factor is enough, its DNA was damaged, and is controlled by a complex of proteins, in which the protein 53 and the CKS cyclin DE play a role. For example, in the G1 to S phase, in the start of G1, the concentration of cyclin was increased. And also in the middle phase, the cyclin E was increased until it reached its maximum point. So if it reached its maximum point, this is called the signal protein for the cell to start change from the G1 phase to S phase and start to synthesize the DNA. Otherwise, if the DNA was damaged, the nutrient is not enough, it will, the cyclin E was not enough here, it will turn to the resting state, the G0, and slow down the cell cycle of the G1 phase, elongate the G1 phase to prepare again the nutrient and all the other factors. The second checkpoint is the G2 checkpoint here, or sometimes we call it the G2 to N, because it must change between the G2 and the M phase. So this one is checking for the cell side was enough to turn into the mitotic phase. If the cytoplasm is enough, the ribosome, the mitochondria, all the ingredients is enough prepared for cell division. Because in this stage, the DNA was already replicated, so it's not checking for the DNA replication was finished, so we check if the DNA replication will have any error happening during the cycle. So this checkpoint was controlled by another cyclin, which is cyclin A. This cyclin A was interacts with the protein kinase CDK1. So kinase is a group of family member enzyme that phosphorylate the histone during the process. So if the cyclin A will reach in the certain point, so it's a signal that the G2 is going on the plan, and the cell will start the checkpoint to start going to the mitotic phase. And the last checkpoint is the M checkpoint, which is happen in the late phase metaphase. And this is also called the spindle checkpoint, to examine that the sister chromatid was correctly attached to the microtubule and separate equally into two corners of the cell and form two different daughter cells. Of course, although we have in the cell cycle checkpoint, sometimes it still makes an error, right? We still have a disorder related to the failure in separating during mitosis and meiosis, like we have the Down syndrome, We have Turner
Có mèo
syndrome
được
and we have Klinefelter syndrome.
thiên ngu ấy,
But we will go to it
trong
later.
ngoài ngu tí.
So
Đây.
in the end checkpoint, we
Wow,
will
not
have the most the most popular corresponding protein that regulate this checkpoint is the cyclin B, which have the interaction with CDK1. So in the end, in the beginning of mitosis, the cyclin B will calculate if the spindle checkpoint was passing through, and if passing through, the cyclin B was degraded by the CDK1 and signal the cell that the mitosis is happening normal.
Cell growth and division are divided into phases. Cells grow during the G1 phase, and the genome replicates during the S phase. During the G2 phase, cells continue to grow and prepare for cell division. During the M phase, mitosis occurs. Cytokinesis, C, is the phase when the cytoplasm divides, creating two daughter cells. Three principal checkpoints control the cell cycle in eukaryotes. The G1 checkpoint makes the key decision as to whether the cell should divide, delay division, or enter a resting stage. The G2 checkpoint assesses the success of DNA replication and triggers the start of the mitosis, M phase. If this checkpoint is passed, the cell initiates the beginning of mitosis. The accuracy of mitosis is assessed at the M checkpoint. This checkpoint occurs during metaphase and triggers the exit from mitosis and cytokinesis and the beginning of G1. At the G2 checkpoint, cyclin-dependent kinases, CDKs, phosphorylate histones and proteins that carry the cycle past the checkpoint into mitosis. During G2, the cell gradually accumulates G2 cyclin, also called mitotic cyclin. The cyclin binds to CDK to form a complex called MPF, mitosis promoting factor. When the level of MPF exceeds the threshold necessary to trigger mitosis, the G2 phase ends and mitosis begins. One of many functions of MPF is to activate proteins that destroy cyclin. As mitosis proceeds to the end of metaphase, CDK levels stay relatively constant, but G2 cyclin is degraded, causing progressively less MPF to be available and initiating the events that end mitosis. After mitosis, the gradual accumulation of new cyclin starts the next turn of the cell cycle. The G1 checkpoint is thought to be regulated in a similar fashion. The level of G1 cyclin increases and associates with cyclin-dependent kinase, CDK. Eventually, a threshold ratio that triggers the next round of DNA replication is reached. The cyclin is degraded and the cycle begins again.
So, come to the cell cycle. So the stem cell is, let's say, one of the most fastest the cell that has the fastest cell cycle in the body. We have a cell cycle just 24 hours. I mean, after 24 hours, we will divide one. But when it's differentiated, it will come to unipotent stem cell, because we will learn more about different stem cells in chapter 7. But you need, for the moment, just know that unipotent stem cell is a stem cell that just can be differentiated to a certain type cell. For example, the unipotent stem cell who produce muscle cell can only produce muscle cell. It cannot produce blood cell, it cannot produce brain cell, it cannot produce skin cell. Meanwhile, the polypotent stem cell can differentiate it into different cell types. Okay? Anyway, in here, most of the cell line have the S phase, the G2 phase, and the N phase. That means the time to replicate DNA in S phase. G2 is preparing for mitosis, and the N phase, that means the mitotic phase, the device phase, is the duration more or less are the same, which is about 12. However, if the length of the G1 phase is different in most cells, which is causing the cell type, the life cycle of cell type is very bright. For example, the intestine, the cell in our gut, have a cell cycle of just about 4 to 5 days. Meanwhile, the liver cell have the life cycle from 300 to 500 days, and the muscle cell have a life cycle of around 16 years. So it come up to a question. In the medical history, we have many types of cancer. We have liver cancer, we have skin cancer, we have prostate cancer, we have brain cancer, but we barely, I will say that we never evidence, but we barely have the muscle cancer or the heart cancer. Do you know why? Yeah, please.
Thì cancel là cái việc mà nó phân ra tế bào không, mà cái này loạn thì nó sẽ không loạn đỡ đấy đúng không? Một lần đỡ vẽ rồi. Đấy.
It's one of the hypotheses, yeah. So right now, science don't know exactly why we barely have heart cancer or muscle cancer. But that's based on several hypotheses. The first hypothesis, like Cedar Chase, says that normally the heart cell, muscle cell, have a life cycle very long. So it will divide less than the other cell. And when it's divided less, it makes less mutation, right? Because the more DNA will replicate, the more cell divide, the more mutation it accumulates. The second hypothesis is muscle cell, or especially heart cell, is located in the body part that hardly have any contact with the toxics. For example, like the lung cancer. The lung cancer is keep contact with the environment era, the pollution, the smoking, all the pollution era. So it will have more damage than the other cell. Our liver cell is contact with all the nutrient, intestine cell contact with the nutrient and all the poison that contaminate in the food, in the nutrient. So it will have more damage than the other one. But the heart cell is located where it contact only with the blood. Hardly toxic, something toxic there. So it have accumulate less damage than the others. One more theory is the heart cell is an important stem cell. So it's all of the part most of the gene was inactivated. Only activated part of the gene related to its function. So all of all the accumulating mutation was benign, right? Because the mutation only harmful when it's expressed out. It cannot be cause any harmful when the gene containing the mutation was silent. We'll come to it in chapter 7. But that is a few hypotheses to explain why we hardly have a cancer in heart cell or in muscle cell. So the cancer, normally all cancer is come from DNA mutation. Some is come from the germline mutation, which is passing through generation. That means you inherited that mutation from your parents. Some mutation was somatic cell that you accumulated in not a gamete cell, in a somatic cell when you have an impact of environment which make a mutation. But so it also starts with a cell or group of cells that not follow the order of the body. So in cell we have a mechanism called apoptosis. Apoptosis is cell death programming. So this apoptosis is when a cell was having to maintain its function, and one is receive a signal that it has to die. The cell will destroy itself through apoptosis. But with some cell, there's a problem with this apoptosis, and it leading to its continuing divide uncontrollably until it forms a tumor. A group of tumors have the arrow just like the first one. So you can consider that this cell is like a stem cell because it's divided uncontrolled. So when it's reached the tumor is large here, that it reaches the bloodstream, and the uncontrolled, undivided cell follow in the bloodstream, forming to travel in the bloodstream in the other region of the body is called a metastasis phase. Okay. So the secret is, under certain time, we all have the cancer cell in our body. So you can see here in the title. Because come along with time, we accumulating mutation. Of course, the mutation rate is depending also in lifestyle. If you smoke, if you drink, if you stay late during the night, the mutation rate will be faster. But faster or later, we also have DNA mutation in our body, and we have all cancer-like body. Okay. However, most of us, those cancer cells cannot be developed because it was detected and destroyed by our immune system. The immune system detects that this cancer cell in our body, like right now in my body, there are some cell was cancer cell. It keep dividing, and my immune system hopefully detect it and destroy it on time. In several cases, when the immune system was weak, it cannot detect the cancer cell. So this cancer cell have to change to develop and start to develop into the cancer tumor. So knowing this, we have developed a therapy to treat cancer called immunotherapy. So beside of the chemical therapy, beside of the radioactive therapy, we have also immunotherapy. So this one have been applied in the patient when they have cancer. The doctor, the medical doctor, have inject them with infectious disease that normally occur. For example, that virus. As they have an infectious factor inject in their body start to react against the infected, like the fever, to kill the virus, to kill the infectious disorder. And scientists find out that when the fever came down, not only the infectious factor, but also the cancer tumor would decrease size. So that means that during the fever, okay, the immune system was enhanced and increased its rate to kill not only the infectious factor, but also kill the cancer cell, which is decreased into the size of the cancer tumor.
Sao không để cúm mãi?
Nếu mình nhớ không nhầm thì ở bệnh viện Vinmec người ta cũng đang phát triển cái chương trình, với lại cái liệu pháp này gọi là
Lấy cúm chị
liệu pháp miễn dịch điều trị ung thư, nghĩa là người ta quan sát thấy khi mà một số bệnh nhân, đa số các bệnh nhân ung thư trẻ tuổi, khi mà người ta bị ung thư, người ta mắc thêm một cái bệnh truyền nhiễm nào đấy, ví dụ như là bị sốt vàng da, hay là bị sốt xuất huyết, hay là bị một cái bệnh truyền nhiễm nào đấy, thì như một phản ứng tự vệ thôi là cơ thể sẽ cái hệ thống miễn dịch cơ thể sẽ được cường hóa, đúng không ạ? Và bệnh nhân sẽ có những cơn sốt. Những cơn sốt đấy thì khi mà cơn sốt hạ đi, và tự cơ thể bệnh nhân làm hạ cơn sốt đấy đi, là cái hệ miễn dịch đấy không những tìm và tiêu diệt cái tác nhân xâm nhiễm vào, cũng như là virus hay vi khuẩn, mà còn kích hoạt cả cái cơ chế giết các cái tế bào ung thư, dẫn đến việc là các cái khối u của những bệnh nhân đấy nó nhỏ đi. Tuy nhiên ở những bệnh nhân này thì khi người ta phát hiện ra là khi bệnh nhân mà sốt quá không chịu được thì hay sử dụng cái thuốc paracetamol. Paracetamol là cái thuốc rất là phổ biến để điều trị cơn sốt, khi mà làm có tác dụng giảm đau này, hạ sốt nhanh. Thì ở những bệnh nhân này hạ sốt này thì cái tác dụng làm cái hệ miễn dịch là giết các tế bào ung thư nó không còn được như cũ. Có nghĩa là khi cơ thể kích hoạt cái đáp ứng miễn dịch thì nó phải gây sốt. Khi mà cái paracetamol đấy làm giảm tác dụng sốt thì đồng thời làm giảm luôn cái tác dụng của cái điều trị miễn dịch khi mà điều trị lại bệnh ung thư, cũng như là cái tác nhân miễn dịch đấy, cái tác nhân xâm nhiễm đấy. Thế thì ở bệnh viện Vinmec bây giờ thì mình biết là có cái chương trình là người ta lấy các cái tế bào sinh miễn dịch của bệnh nhân ra, nuôi nó lên, cường hóa nó, sau đấy lại bơm trở lại cơ thể để nó có thể điều trị bệnh ung thư. Mà tác dụng đến đâu thì họ không công bố nên mình cũng không biết. Mình chỉ biết có chương trình như thế thôi.
Đập thẻ ấy nhỉ? Kiểu đập thẻ.
Alright, so the cell cycle is leading to the corresponding, the wondering in scientists that which kind of genes that most related to cancer. And after a couple of decades, researching in many cancer type, they found out that the most two gene BRCA1 and BRCA2, especially BRCA2, is related to many cancer types. For example, breast cancer or lung cancer. So these two genes was encoding for a protein that involved in the DNA repair mechanisms and corresponding to the protein that controlling the G2 and M cell cycle checkpoint regulation. So we can see that if we have any mutations causing in these two genes, it will lead to the protein encoding by the two genes was missing its function, lead to consequences that all the cell cycle checkpoints was defective. So all the regulation to control the cell cycle and to put the cell under the resting place like the G0 or apoptosis was failed. And as a consequence, the cell was divided uncontrollably and developed into the cancer tumor. Okay. Alright, so I think our session today will be finished here. Do you have any questions? Okay, thank you very much and see you in Friday.